Hello, my name is Josh Mugabe. I welcome you to Coordination and Control, questions 16 to 20. (16) Explain what is meant by the following. 1. Visual acuity. Number 2. Retinal convergence. Part B. Explain how the mammalian eye achieves each of the following. Number 1. Visual perception under dim light. Number 2. Color perception. Explain what is meant by the following. Number 1. Visual acuity. Visual acuity, this is the sharpness or clearness of vision. It depends on the sharpness of the retinal focus. Visual acuity is done by the cones, maximally at the fovea. It involves the ability of the eye to distinguish between objects that are close together, as well as accurate distance judgment. Each cone synapses with a single bipolar neuron. Each generates its own receptor potential above threshold, sends separate impulses to the brain, which interprets two or more images partially separated. So we are looking at the cones. We have here, this is our cone cell. And when you look at that cone cell, we are saying that each cone cell synapses with a single bipolar neuron. So when you come here and you look at the cone cell, here it forms a synapse. And this synapse, it is one cone synapsing with one synapse. There is only one synapse alone, one cone and one synapse, and it will afterwards get these impulses. They will be sent to the brain through a single bipolar cell or bipolar neuron to the brain, and then it will be interpreted, and then a clear image will be observed. Let's now look at retinal convergence. Retinal convergence. Retinal convergence, this involves the rod cells establishing one synapse with a single bipolar neuron, which also connects to a single nerve fiber within the optic nerve. Sensitivity to low light intensity is achieved this way. Each rod cell stimulation establishes its own receptor potential. When you talk about retinal convergence, we are talking about the rods. Here we have 3 rod cells, and each rod cell has got a synapse. But when these synapses here, when they send the impulses are going to be joined together. When they create a receptor potential, they join together and connect to one bipolar neuron, one single bipolar neuron. And then here, all the information will be carried by a single neuron to the brain, which information is coming from all these three rod cells. Several receptors potential from different rods spatially submit, form one large receptor potential whose magnitude is beyond threshold, creates an action potential, fires an impulse to the brain, which interprets one image. So when you look at our three roads, and each one is going to, its receptor going to produce a receptor potential. Each one will produce its own receptor potential. But these receptor potential are going to sum up and they will form one large receptor potential, which can be beyond the threshold. Now, when it is above the threshold, an action potential is going to fire an impulse, which is going to be passed through one bipolar neuron, and it will be transported to the brain. And the brain is going to interpret it as a single image, yet it came from three rod cells. But because of the summation here, combining spatial summation to form one receptor potential, it is recognized as one image. Part B. Explain how the mammalian eye achieves each of the following. Number one, visual perception under dim light. At low light intensity, this evokes the parasympathetic discharge, which causes relaxation of the papillary sphincter muscles, those are the circular muscles, and a contraction of the dilator papillae, these are the radial, resulting in papillae constriction. So here we have the eye. We have our eye here, and we have the iris. This is our iris muscles. The ones inside are circular muscles, and the ones outside here, those are radial muscles. So under low light intensity, the parasympathetic is going to stimulate the circular muscles to relax. And when they relax, they will be moving outwards, going out like that. That means that they are relaxing. And when they are relaxing and moving outwards, That means that now these radial muscles are contracting. So the radial muscles, as they are contracting, they will be pulling the circular muscles outwards. So the radial muscles will become shorter as they are pulling. There are circular muscles outside. They are contracting. And when they contract, what is going to happen is that now this gap here, which we call the pupil, is going to become wide. And when it becomes wide, it is going to allow a lot of light to enter into the eye. So the amount of light entering the eye increases. The light gets diffracted by the cornea and the lens through the vitreous humor, and it strikes the rods of the retina. The rhodopsin converts to opsin and all-trans retinol. This closes the sodium-mediated protein channels, and the influx of sodium ion is reduced in the outer segment of the sodium pump. In the inner segment, continues to pump out sodium ions. So when the light reaches our cell, the retinal cell, the rhodopsin is going to change into opsin and all trans retinol. And this one is going now to close the sodium mediated protein channel. So sodium will not be allowed to enter into the photoreceptor, the rod. It will not be allowed. That is in the outer segment. Now, in the inner segment, what is going to happen? The pump, which we call the sodium pump, is going to continuously pump out. It will be pumping out the sodium ions out of the cell. So when the sodium pump is pumped out in the inner segment and in the outer segment, the sodium ions are prevented from entering. Inside, it is going to become more negative. Rod cells get hyperpolarized, and the rate of release of the excitatory neurotransmitter is released at the inhibitory synapse. The adjoining bipolar neurons also get hyperpolarized, but the ganglion cells of the optic nerve supplied by the bipolar neuron respond by producing a receptor potential. So what we are having, our rod cells are going to be hyperpolarized, and at the excitatory neurotransmitter is going to be released in the inhibitor synapse, and the adjoining bipolar neuron also gets hyperpolarized, but the ganglion cell here down, this one is going now to generate its own receptor potential. This partial summate establish a receptor potential beyond the threshold that creates an action potential that fires impulse to the brain. So as we have seen, the other side, 3 rod cells, they summate up their receptor potential to create an action potential which is firing an impulse to the brain. Let's now look at color perception. Color perception, this one is achieved by the 3 isoform of cones. There is the red, the green, and the blue cones. Iodopsin, the photosensitive pigment in the outer segment, occurs in 3 isoforms, each occurring in one of the 3 cones, sensitive to either red, green, and blue light. So the iodopsin, which is the photosensitive pigment, is found in the 3 isoforms. One in the blue cones, the green cones, and the red cones. Light of a particular wavelength, on stimulating the responding cone, bleaches the corresponding isoform of iodopsin and establishes an action potential. Information is relayed to the brain, which interprets the color corresponding to the wavelength of the stimulant light 100%. Sole stimulation of green, red, and blue cone leads to perception of the green, the red, and blue colors respectively. Equal stimulation of the red and green cones, the yellow light or yellow color is perceived. Equal stimulation of red and blue cones, magenta is perceived. Equal stimulation of green and blue cones, cyanine is perceived. If all the 3 cones are equally stimulated, then the white light or color is perceived, whereas black is perceived when none of the cones is stimulated. Other colors are perceived based on the relative stimulation of the 3 colors. Number 17. Distinguish between photoperiodism and photoperiod. Part B. State the effect of photoperiod on the dormancy of plants. Part C. Describe the role of phytochrome system in the control of flowering in plants. Distinguish between photoperiodism and photoperiod. Photoperiodism is the biological response of an organism to the relative changes in the length of the day, while photoperiod is the relative length of the day. State the effects of photoperiod on the dormancy of plants. Photoperiod greater than the critical period, which is 12 hours. This one is going to promote flowering in long day plants and day neutral plants, but not short day plants. Whereas photoperiod less than the critical period, it will promote flowering in short day plants and day neutral plants, but not long day plants. We are still looking at the effect of photoperiod on the dormancy of plants. We are now looking at seed dormancy. Photoperiod greater than the critical period, it promotes the breakage of seed dormancy. While photoperiod less than the critical period promotes seed dormancy. What about bud dormancy? Photoperiod greater than the critical period breaks the bud dormancy, and the photoperiod less than the critical period, it promotes bud dormancy. Part C. Describe the role of phytochrome system in the control of flowering in plants. Phytochrome system consists of two interconvertible proteinous molecules, phytochrome red and phytochrome far red. Phytochrome red is more stable in far red light. Thus, its concentration is more abundant at night. while phytochrome far red is more stable in red light. Therefore, its concentration is more during the day. Phytochrome red favors flowering in short day plants, but inhibits it in long day plants. Phytochrome far red favors flowering in long day plants and inhibits it in short day plants. During the day, presence of plenty of red light causes the conversion of phytochrome red to phytochrome far red. Phytochrome far red induces the release of florigen, stimulating the dormant buds of long day plants to develop into flowers. In short day plants, subjecting the plant to longer hours of the night and shorter days enables the slow conversion of phytochrome far red to phytochrome red. Phytochrome red stimulates the release of florigen that activate dormant buds to bloom into flowers. Number 18. What is meant by vernalization? Part B. Explain the effect of light or darkness interruptions on the flowering in angiosperms. Part C. Explain geotropism in plants. Explain what is meant by vernalization. Vernalization refers to the exposure of the plant to low temperature requirements that allow initiation of flowering. The buds would stay dormant until exposed to the temperature as low as 4 degrees Celsius. These temperatures stimulate the release of the hormone vanillin from the shoot apex. It travels to the dormant buds, causing them to develop into flowers. The hormone stimulates the secretion of the growth promoters like gibberellins, which potentiate the development of buds into flowers. What is explain the effects of light or darkness interruption in flowering in angiosperms? We shall start for short night and long days. Interruption of the short night with brief periods of white light has no effect on flowering. Flowering occurs because the interruption causes shorter nights that maintains a higher concentration of stimulatory phytochrome far red. Compared to inhibitory phytochrome red, since phytochrome far red to phytochrome red conversion is slow and requires exposure of the plant to a longer night. Interruption of the long day with brief darkness does not affect flowering because still higher concentration of stimulant phytochrome far red compared to inhibitory phytochrome red. Interruption of long days with long periods of darkness. Flowering fails due to conversion of stimulatory phytochrome far red to inhibitory phytochrome red, such that phytochrome red concentration greatly exceeds that of phytochrome far red. Let's now look at shorter day and long night plants. The effect of light or darkness interruption on the flowering. Now, interruption of the long nights with brief periods of white light, no flowering occurs because interruption causes rapid conversion of stimulatory phytochrome red to inhibitory phytochrome far red. Besides, interruption splits the longer nights into shorter nights. The inhibitory effect of flashing white light in the middle of the night is reversed by providing longer interruption with far red light. What of the day neutral plants? Flowering is not affected by any form of light or darkness interruptions because flowering in this plant is independent of the photoperiod. Explain geotropism in plants. Geotropism is the plant's movement to unidirectional gravitational force. The plant's shoot is negatively geotropic, while the roots are positively geotropic. Geotropism is controlled by auxin. These are growth substances that mediate differential growth. During germination, when a horizontally lying seedling is subjected freely to gravity, auxins are made to accumulate on the lower side of both the radicle and the plumule. A high concentration of auxin on the underside of the plumule causes differential growth, in which there is more growth on the underside, causing upward curvature of the shoot. On the other side, growth is inhibited. On the underside of the radicle, causing downward curvature of the root. Geotropism is also attributed to Statorith, special starch granules whose free fall under gravity initiate geotropism. Increase in temperature decreases the time of fall of the Statorith. Compare positive and negative feedback mechanism of homeostasis. B. Explain the role of positive feedback in nervous transmission. Part B. In parturiation. Part C. Explain how the deviation in estrogen levels in the blood reduces to normal. Part A. Compare the positive and negative feedback mechanism of homeostasis. Let's start with the similarities. Both feedback mechanisms, they are triggered by a deviation from the normal physiological set-up point. Both feedback mechanisms involve joint activities of the detector, controller, and effectors. Let's now look at the differences. In positive feedback, collective mechanisms continuously amplify the deviation from the physiological set point. In negative feedback, a corrective mechanism nullifies or stops further deviation from the normal. In positive feedback, the aim is to potentiate further deviation from the normal. In negative feedback, the aim is to restore back the physiological set point which is the norm. In positive feedback, detectors always switched on, recognizes deviation, the deviation, and continuously alert the controller. In negative feedback, detector gets switched off such that the controller is no longer alerted of the deviation. In positive feedback, it is few in biological systems. Negative feedback is very common in biological systems. In the positive feedback, examples include the clotting cascade, oxytocin release, nerve impulse transmission. Negative feedback examples include blood sugar regulation, regulation of hormones like anterior pituitary hormones, enzyme activity, and many others. Explain the role of positive feedback in Roman number 1, nervous transmission. Propagation of impulses causes cancellation of the negative resting potential at the point that is complementarily depolarized, making the axon membrane potentially positive. The sodium gates, being sensitive to depolarization, permit more sodium channels to open and increase sodium ion influx. As sodium ions pass through the axon, through the membrane, they cause further depolarization. And depolarization and sodium channel opening keep reinforcing and amplifying each other, constituting a positive feedback loop. Let's now look at parturition, the role of positive feedback in parturition. Head of the fetus pushes against the cervix. Nerve impulses from the cervix are transmitted to the brain. The posterior pituitary gland is stimulated to secrete oxytocin, which is carried through the bloodstream to the uterus. Oxytocin causes release of prostaglandins that initiate uterine contractions, causing the fetus to push further towards the cervix. And this further amplifies more oxytocin release, coupled with subsequent uterine contraction. Explain how the deviation in estrogen level in the blood is reduced to normal. Estrogen levels are regulated by negative feedback mechanism by the hypothalamus pituitary gonadal axis. High levels of estrogen suppresses the synthesis of kisspeptin, a stimulatory protein that activates the release of gonadotrophin-releasing hormone from the hypothalamic neurons. The levels of gonadotrophin-releasing hormone thus reduce, and this inhibits the release of the gonadotrophins, that is follicle-stimulating hormone and luteinizing hormone, from the pituitary gland. Follicular growth of the ovaria and eventual ovulation ceases. Release of estrogen from the thicker and granulosa cells of the Graafian follicle is finally terminated until the physiological set point is re-established. Number 20. Define the term generator potential. Part B. Describe the behavior of the receptor cells in absence and presence of an appropriate stimulus. State the four structural differences between the two main branches of the autonomic nervous system. Define the term generator potential. Generator potential refers to the membrane potential established at the sensory cell after its local depolarization in response to the graded strength of a stimulus applied to the associated receptor. Part B. Describe the behavior of a receptor cell in absence and presence of an appropriate stimulus. At rest, potassium channels open, while sodium channels close. Thus, potassium freely diffuses from the oxoplasm to the extracellular fluid without reciprocal entrance of sodium ions. Sodium-potassium pumps out 3 sodium ions in exchange to 2 potassium ions. The net effect is a higher cationic concentration on the outer axonal membrane relative to the inner, making the membrane positively charged on the outside relative to inside. The membrane gets polarized and the resting potential is established. No impulse is transmitted to the sensory neuron. Upon stimulation, the sensitive parts of the receptor cell there is a reversed permeability of axon membrane to sodium and potassium ions. The sodium channels open and the potassium channels close. Thus, the membrane becomes more permeable to sodium ions than potassium ions, causing an influx of sodium ions into the inner membrane along the electrochemical gradient. Inner axonal membrane therefore becomes more positive relative to the outside membrane. The membrane gets depolarized. Established membrane potential difference constitute the receptor and the generator potential. A subliminal or subthreshold stimulus fails to evoke an action potential, while a threshold stimulus establishes an action potential, builds to the threshold, creates an action potential, fires an impulse across the axonal membrane of the sensory neuron. Part C. State 4 structure differences between the two main branches of autonomic nervous system. Sympathetic nervous system. It has got a short preganglionic fiber. Parasympathetic nervous system has got a long preganglionic fiber. So let us try to see here. This is our central nervous system, and this one is the parasympathetic nerve system, and this one is a sympathetic nerve system. This circle we are seeing here, that is a ganglion, and this one is also a ganglion. Now there are fibers that connect. This fiber of the parasympathetic here is called a preganglion fiber, and this fiber here connecting to the ganglion of sympathetic is also called a preganglion fiber. So for the sympathetic neuron, you can see that the preganglionic fiber is short compared to the preganglionic fiber of the parasympathetic nervous system. Here we are saying that in the sympathetic nervous system, they have a long postganglionic fiber, and the parasympathetic nervous system, they have a short postganglionic fiber. And now if we look here, we said that this is our ganglion of the sympathetic nervous system, and this is also a ganglion of the parasympathetic nervous system. So now here, this fiber here is the postganglionic fiber of the sympathetic, and this one, this side, from here going to the effector, is the postganglionic fiber of the parasympathetic nervous system. So you can see that the postganglionic fiber of the sympathetic nervous system is longer than the postganglionic fiber of the parasympathetic. Sympathetic autonomic ganglion is close to the central nervous system and far away from the effector. For parasympathetic nervous system, the autonomic ganglion is closer to the effector organ and away from the central nervous system. So let us come back and look here. We can see that this is our central nervous system. And we have said this one is our ganglion of the sympathetic nervous system, and this one is also a ganglion of the parasympathetic nervous system. Then this side here, this one is the effector, where the sympathetic nerve is going, and this one is the effector where the parasympathetic nerve is going. So when we interpret and we look at them in terms of ganglions, you will see that the ganglion of parasympathetic is very far away from the central nervous system, and it is very close to the effector. Whereas if you look at the ganglion of the sympathetic nervous system, it is very close to the central nervous system and very far away from the effector. In sympathetic, emerges from the lumbar and thoracic segments of the spinal cord. In parasympathetic nervous system, it emerges from the brain or cranial nerves and few from the sacrospinal segment. So when you are comparing the spinal cord where the parasympathetic is coming from, it comes from two regions: the thoracic region, the spinal cord of the thoracic region, and the spinal cord of the lumbar region. Whereas if you talk about the parasympathetic nervous system, it comes from the brain part or the cranial nerves, and then the spinal cord part of the sacral segment. That's where the parasympathetic nervous system emerges from. Parasympathetic nervous system, numerous postganglionic fiber. They have numerous postganglionic fibers. In parasympathetic system, there are few postganglionic fiber. So the postganglionic fibers are these ones that are coming from the ganglion. For the sympathetic, there are very many fibers of the postganglionic fiber. Whereas for the parasympathetic, there are very few postganglionic fibers. in sympathetic nervous system, noradrenergic receptors at the effector organ, whereas the parasympathetic nervous system, it is cholinergic receptors at the effector organs. We have said here our effectors are these ones. This is the effector, and this one is also an effector. And here we have the parasympathetic nerve, and the other side we have the parasympathetic nerve. Now, at the point where they communicate with the effectors, their synapse, and at those synapse, for the sympathetic synapse here, it releases a transmitter substance called noradrenaline. So the receptors in the effectors will receive noradrenaline, so they refer to them as noradrenergic receptors. Whereas the synapse here of the parasympathetic to the effector here, it releases a transmitter substance called acetylcholine, and therefore the effector has got receptors which we call cholinergic receptors. They receive acetylcholine. Thank you very much for participating in coordination and control, questions 16 to 20. Please do like, share, and subscribe.